Thank you very much for coming this Saturday morning. Today's guest speaker is Dr. Laura Ranum, and she's a Kitzman Family Professor of Molecular Genetics and Microbiology, and the founding director of the Center of Neurogenetics at the University of Florida. In 2011, her lab overturned long-standing dogma in the field by discovering that an entire class of novel proteins are produced from repeat expansion mutations without any canonical AUG initiating codons. And these toxic repeat-associated non-AUG run proteins have been shown to accumulate in a growing number of neurodegenerative diseases, which include Huntington's disease, amyotrophic lateral sclerosis, frontotemporal dementia, and many others. Mechanistic insights from her recent work have identified novel therapeutic strategies to treat RUN protein disorders. Dr. Ranum's scientific contributions have been honored by multiple awards, which include Hans Steinert Award for meiotic dystrophy research, election as a fellow of the American Association for Advancement of Sciences, Javits Neuroscience Investigator Award, NIH NINDAs and Biology, and additionally, she served as a co-chair of 8 international scientific meetings and is advocate for ataxia meiotic dystrophy and ALS patients. I don't want to keep Dr. Ranum away from her seminar anymore. It's a great pleasure to have you in our ZOOMinar series, it's your turn. Thank you very much for coming.
Thank you very much for the kind introduction and for the invitation. I'm really happy to be able to present this work to you today. So I'm going to give you an overview of RAN translation, a little bit of history, and then I'll talk about the mechanisms of RAN translation and then our recent work in targeting RAN proteins in C9orf72 ALS FTD BAC transgenic mice. So microsatellite expansion diseases, when they were first identified in about 30 years ago this year, they were thought to have 3 primary mechanisms. That is, if the repeat expansion was in an upstream region, like the CGG expansion mutation for Fragile X mental retardation, it was thought to cause protein loss of function because it shut down the expression of the transcript, and there was little FMR1 protein. If the expansion mutation fell within an ATG-initiated open reading frame, an example is Huntington's disease, which makes a polyglutamine expansion. Those diseases were thought to cause disease by protein gain of function effects. And myotonic dystrophy for many years was quite a puzzle because it's a dominant disease, but the repeat expansion mutation is located in a non-coding 3' untranslated region. And subsequent work led to the realization that this RNA can sequester RNA binding proteins in the nuclei of cells and cause abnormal splicing and regulation of RNAs. And so that's an RNA gain of function effect. And this is how the field of microsatellite expansion mutations has sort of was anchored for many years. And so as the years went on, more and more genes were identified and they kind of fell into these categories. There were intronic mutations, myotonic dystrophy type 2, 3' untranslated, genes with 3' untranslated expansion mutations, and more polyglutamine encoding repeat expansion disorders. But things started to change a bit in 2005 and 2006. Stephen Tapscott's lab in 2005 showed that the myotonic dystrophy type 1 repeat could be bidirectionally transcribed And in 2006, we showed that the SCA8 expansion mutation can be bidirectionally transcribed. And for SCA8, there was a funny little polyglutamine protein that was made that wasn't really part of a larger open reading frame. And we cloned the gene for spinocerebellar ataxia type 8 some years ago, and we had been, because it causes a limb and gait incoordination, eye movement abnormalities, slurred speech, cerebellar degeneration. But a very puzzling part of the disease is that it didn't cause disease in everybody that had the expansion mutation. So, but it could run in families, and and affected individuals had marked cerebellar degeneration, which you can see on the MRI, but the reduced penetrance really made this a controversial discovery. And because of the segregation in a large family that we had been studying, and because we cloned this from a single affected patient and then found it in a number of ataxia families, we really didn't think that we could have come up with this just by chance alone. And so we went on to make a BAC transgenic mouse model. And this is the gene as we understood it at the time, that the repeat expansion was in a 3' untranslated region of a long non-coding RNA. We made a mouse model of the disease, and the mice developed neurological phenotypes and had cerebellar abnormalities, so we were excited about that. This expansion mutation, because it was located in a 3' untranslated region, we expected it to maybe have a mechanism similar to what was discovered for myotonic dystrophy type 1, which was an RNA gain of function mechanism. And we have evidence that there are RNA foci that accumulate in the mice and for RNA gain of function effects in our mice. But we also discovered that there was a polyglutamine protein, and that polyglutamine protein accumulates in both the mouse model and in human tissues. And so that was very exciting to us, because that meant that now we had a pathologic link between the repeat expansion mutation and the disease. And the polyglutamine expansion mutation came from a transcript that we didn't expect to find, which was expressed in the opposite direction. And the polyglutamine open reading frame appeared to be this pure polyglutamine protein with stop codons right after the polyglutamine track and a single AUG initiation codon upstream. So we were really focused on trying to understand what's more important, the RNA gain of function effects or the polyglutamine in SCA8. And we did a number of experiments and a number of control experiments, and that led to an experiment in which we mutated this ATG initiation codon to an AAG. And Tao Zhu did this experiment and found a very surprising thing, which we thought initially was an error, in that the polyglutamine protein didn't go away. And so after repeating this experiment, checking the sequence, and becoming convinced that this was really happening, we started to ask the question, well, can you make polyserine and alanine if you don't need an AUG initiation codon? What even sets the frame? And the answer was, yes, you can make polyglutamine, polyalanine, and polyserine from a CAG expansion mutation without an AUG initiation codon. And he tagged each of these proteins with epitope tags in all of the reading frames. And this phenomenon seemed to be dependent upon RNA structure. So CUG repeats and CAG repeats did this, but CAA, which doesn't form a hairpin, does not do this. And we had a lot of trouble having this idea accepted. So we did many control experiments, and one of them was to do mass spectrometry and show that we indeed did have a polyalanine protein being expressed. And then we went on to show that these proteins can accumulate in vivo. So in both the mouse and the human autopsy tissue, we could find evidence for a polyalanine protein by immunohistochemistry, and also for a polyglutamine protein being expressed from myotonic dystrophy type 1, the mutation from myotonic dystrophy type 1, which is a CUG expansion in what was thought to be a non-protein coding region of the gene. And actually the polyglutamine would be made from the opposite strand CAG repeat, but also without an AUG initiation codon. So this process we called repeat associated non-AUG translation. And now this has been shown to occur in a whole bunch of different diseases, including by Peter Todd's group for Fragile X tremor ataxia syndrome. But a large number of diseases now have been shown to make these RAN proteins, and they can be homopolymeric proteins, like in Fragile X tremor ataxia syndrome, dipeptide repeats, pentapeptide repeats, tetrapeptide repeats. And, I expect that more and more will be found as we go along. There are 50 microsatellite expansion diseases and now 11 diseases that have shown evidence for RAN proteins accumulating in the patient tissue itself. So I'm going to focus most of the rest of the talk on C9orf72 ALS FTD. This expansion mutation was very exciting for the field, the repeat expansion field, and also the ALS field and the dementia fields, because it was the first time that a repeat expansion mutation had been shown to cause ALS and frontotemporal dementia. And it kind of led to a confluence of investigators that were very interested in understanding how this worked, and a real infusion of talent and efforts to understand the molecular mechanisms of microsatellite repeat diseases. So this expansion mutation is the most common genetic cause of ALS and FTD, and variations in the occur in the onset and the disease presentation. So it can cause ALS or frontotemporal dementia. And it also has this reduced penetrance that we saw in SCA8. So not everybody with the expansion mutation by the time they're 80 develops C9orf72 ALS FTD. This expansion mutation, similar to some of the other work on other expansion mutation, is also bidirectionally expressed. So there's a G4C2 expansion in the sense direction and a G2C4 expansion mutation expressed at the RNA level in the antisense direction. And there are 6 different dipeptide proteins, 5 different repeat motifs, but 6 unique proteins if you take into account the C-terminal ends, which are unique. And we and others have shown that these proteins can accumulate in human autopsy tissue. And many people study the toxicity of RAN proteins now, and especially the toxicity and molecular effects of the C9orf72 RAN protein. So there's a lot of research activity in this area. And it's clear that these proteins can be toxic when overexpressed in cells, in flies, and in mice. And there are many more studies than listed. But we were really interested in developing a model to understand the contribution of RAN proteins expressed in more of an endogenous setting. And so to do that, 2 graduate students in my lab teamed up to develop a BAC transgenic mouse model of the disease. They used a human BAC transgene with about 100 kb of DNA sequence, so plenty of flanking sequence upstream and downstream of the gene. And they generated several different lines of these mice, which showed phenotypes, including a paralysis phenotype. The affected mice, the acutely affected mice, had really overt neuromuscular junction denervation. There was motor neuron loss that was found in the end-stage mice, and also behavioral deficits that could be found where the mice avoided the center of an open field chamber. In this example, the non-transgenic mice explore the entire cage. The C9 mice tend to avoid the center of the cage, and they can have this phenotype with a motor phenotype where they move less, or where they're just avoiding the center of the cage. These mice also develop the molecular features of the disease, so they express both the sense and the antisense transcripts and the RAN proteins. And so we wondered if we could use this model to prevent disease and to learn about the mechanisms of the disease. And so Dr. Nguyen in my lab, really talented postdoc, took on a project to try to understand if immunotherapy might work. And this was a project in which we teamed up with Neuron, and we looked at human antibodies that targeted RAN proteins. And these antibodies were generated from healthy donor cohorts. And these recombinant antibodies were made, and then what Nguyen did was characterize these antibodies using in vitro validation systems, and then she tested their in vivo validation and their efficacy in our mouse model. And so these antibodies have really high affinity and specificity to RAN proteins. So this is a sub-nanomolar affinity for the anti-GA1 antibody shown here. And then there's very little cross-reactivity with anything else. So anything else that you might suspect would be sticky and might cause an antibody to cross-react, like A-beta, Huntington, the other RAN proteins. So they're very specific and they're very high affinity. And when we express the proteins in cells, they behave much differently. And so we wanted to know if these antibodies will detect the proteins when they're expressed in cells. And of the 20 recombinant proteins that we looked at, we really found 3 that we liked a lot. And those 3 were, could detect the RAN proteins in cells, but also could detect the RAN proteins that were expressed in our BAC transgenic mice. And no similar staining was found in the non-transgenic littermates. So the first experiment that we did to try to understand if this strategy had any chance at all of succeeding was we did a target engagement study in which we used a peripheral injection strategy of the antibodies, and we took mice from 4 to 15 months of age and did a IP injection, and then did a takedown after 2 days or after 10 days. And we found this, we were really excited to see this staining that looked like it could be similar staining to what we saw in the autopsy tissue or in the mouse staining tissue by immunohistochemistry. But we wanted to prove that these were really the RAN protein aggregates, because this is a peripheral injection of a large molecule that then has to cross the blood-brain barrier and somehow get into cells. So it seemed like it shouldn't work, but it seemed like it was working. So to prove that this really could be true, we used a mouse monoclonal antibody that we developed in the lab to the GA protein, and then looked at the animals that had been injected with either IgG as a control or peripherally injected with the GA antibody. And there was a very nice co-localization with these antibodies. Knowing that we could, that these antibodies can get into the brain at some level and co-localize with the aggregates in cells, we decided to move ahead with a larger in vivo efficacy study. And for this study, we injected the mice at 6 weeks of age at a time when they begin to show some phenotypes, including the molecular phenotypes of the RAN protein accumulation. And we used 3 different experimental antibodies, so 2 that target the GA protein and 1 that targets the GP protein. And we used chimeric versions of these antibodies so that they would, chronic treatment of the mice would not cause an immune reaction, an adverse immune reaction. And so the survival curves are shown here, and the non-transgenic, the non-transgenic mice are up here in green, and then the C9 mice that were treated with PBS showed the death curve that we'd seen before. And really, we were very excited to learn that it was the GA antibody that was increasing, one of the GA antibodies that was increasing the survival of one of these groups. And Nguyen and our whole lab was blinded by Neuroimmune as to what these groups were. We just knew that there was a divergence in the group, and we wanted to end the study in time so that we could have tissue to analyze things. So we ended the trial at 43 weeks. But at 24 weeks, these mice that were treated with the GA antibody, GA1 and the GA2 antibody, showed improved behavior by both open field and digigate and cage behavior. And some examples of the digigate are shown here. The GA1 targeting antibody decreased neuroinflammation and neuropathology, a variety of different measures that I don't have time to go into in detail, but these include increasing the motor neuron survival. So in the non-transgenic mice compared to the C9 mice, there's a loss of motor neurons. And in the GA1 antibody treatment group, that is normalized. And a big surprise to us was that the anti-GA1 antibody targeted not only GA, but also GP and GR. And so we were expecting it would target GA, and it did, reduce GA levels when the mice were given the GA antibody. And GA levels are not reduced when the mice were given the GP antibody. And but both GA antibodies worked to some degree, but the GA1 antibody worked better than the GA2 antibody. And the GA1 antibody also reduced the GP levels and the GR levels. So we had this collateral benefit of targeting GA for some reason, decreased the levels of GP and GR as well. So we wanted to know, how does that work? And there's no change in the RNA foci or RNA levels in these antibody-treated mice. We showed it with biochemical studies that the GA antibodies really target the GA proteins. And I'll highlight some of the findings that we had about how the antibodies enter cells. So the glycosylation of the Fc portion of an antibody can be important for cellular uptake with by the Fc receptor. And so we remove the glycosylated portion of this antibody in the Fc region of the antibody. And this would be predicted to bind Fc receptors, but the aglycosylated version of the antibody cannot bind Fc gamma receptors. And we did an abbreviated study in the mice with, again, starting at 6 weeks, but this time we only went to, out to 16 to 17 weeks. And we used a chimeric GA antibody as we had done before, or the aglycosylated version of the antibody, or IgG as a control. And here we show that the glycosylated antibody, as previously shown, reduces the GA aggregates, but the aglycosylated antibody does not. And we showed that, that the loss of neurons in the posterior horn that's seen in the C9 mice that were treated with IgG alone were not reduced when treated with the glycosylated GA1 antibody, but this this did not work. We didn't get a significant improvement using the aglycosylated antibodies. So the glycosylation is important. And then we show that the glycosylation is important for the co-localization in the cells of to the GA aggregates. So glycosylation is needed for cellular uptake, for GA protein clearance, and also for cell survival. So GA1 treatment decreases the GA protein half-life in T98 cells. So for these experiments, Nguyen expressed GFP GA and used an azoalanine methionine analog to track the synthesis of new proteins. And then she measured the protein turnover using a click chemistry to label the proteins with biotin. And she detected the, you can see it's like a pulse chase experiment, and you can see in untreated cells, the half-life of the proteins is about 24 hours. And in the antibody treated cells, the half-life of the protein is 8 hours. So the treatment with the GA1 antibody really decreases the half-life of the GA protein. And if she inhibits the autophagy or proteasome, she found that this decrease in GA levels does not occur. And so the GA clearance by the GA1 antibody is proteasome and autophagy dependent. And then we looked at the possibility that TRIM21, which is a tripartite motif containing protein 21 and an E3 ubiquitin protein ligase, could be an effector molecule that would help get the antibody-bound cargo ubiquitinated and then degraded by the proteasome. And so there's this decrease in the GFP GA levels with the antibody treatment that we see. And when she used the TRIM21 knockdown cells, she did not see the same decrease in these cells that were overexpressing the protein. The GA1 chimeric antibody also interrupts the aggregasome entrapment of other proteins. So the GA forms this network of proteins that traps the proteasome within the GA protein network, but also entraps other proteins. And the antibody treated mice and cells allow this entrapment to be disrupted. And the antibody increases the proteasome activity and reduces the death in cells that overexpress the alternative GA proteins as well. And so this just shows the proteasome activity increases in the presence of the GA treatment, and the GA treatment decreases the percent cell death. And so our working model for the GA effects are that the glycosylation is really important for uptake by the Fc gamma receptor, and aglycosylated antibodies don't enter the cell as well. And once inside the cell, the antibody can bind to the GA proteins, and this likely improves protein homeostasis within the cell. So it prevents blocking of proteasome function, and it likely just increases the turnover of these other proteins and cell survival. That's intracellular, could have extracellular effects as well. So in summary then, for this part of the talk, human anti-C9 antibodies enter the brain and co-localize with the intracellular aggregates. And the anti-GA1 and anti-GA2 antibodies both improve the behavior in our C9 BAC mouse model, but anti-GA1 increased survival and worked better. Anti-GA1 decreases neuroinflammation and neuronal loss in the C9 BAC mice. And surprisingly and really excitingly, anti-GA1 reduced not only the GA levels, but GP and GR proteins also in the BAC mice and in cells. And this really shows then that targeting RAN proteins without affecting the RNA levels is sufficient to mitigate disease, and therefore that the RAN proteins drive disease in our C9 BAC mice. And it also shows that GA protein is a really important therapeutic target, and that you don't have to necessarily target all of the RAN proteins. So maybe it's a simpler equation than we previously thought. So the next part of my talk, I want to switch gears and talk about another therapeutic strategy that we've been working on, which is to turn off RAN protein production. And our rationale is that if we can turn off RAN protein production, perhaps that could be an Achilles heel. Maybe we could get a strategy that would turn off RAN proteins across multiple types of disorders that involve repeat expansions in RAN translation. So what are the mechanisms of RAN translation? So the integrated stress response is a response in which cells respond to different insults, and that leads to the phosphorylation of EIF2 alpha, which is a subunit of the EIF2 protein. And this box is highlighted in green because it was Green and Todd and Chang and Son, but Green was the first author and really showed that EIF2 alpha, when it's phosphorylated, leads to elevated levels of RAN proteins. And we were also looking at this pathway, and Tao Zhu was focused on the PKR pathway and try to make the connection between the hairpin forming RNAs that result from some of these expansion mutations and perhaps stimulation of the viral infection response, protein kinase R response, which will also lead to EIF2 alpha phosphorylation. But you can have misfolded proteins lead to EIF2 alpha phosphorylation, PKR, oxidative stress, nutrient deprivation. There are a variety of different insults that can lead to the activation of this pathway. And if this pathway is activated, EIF2 alpha is phosphorylated, then you get a decrease in global translation and an increase in RAN translation. And when we started this work, we were, as I said, focused on the double-stranded RNA-dependent kinase. This work was really pioneered by Nahum Sonnenberg's lab, who showed that HIV-1 can activate the PKR pathway. but also Charles Thornton's lab showed that expanded CUG repeats that are found in myotonic dystrophy and other diseases like SCA8 can form hairpins that also activate the double-stranded RNA-dependent protein kinase R. And so we wondered if we looked at a variety of different repeat expansions, and in cells have evidence that they can activate phospho-PKR, but these experiments are tricky and depend on transfection and various things. So we really wanted to get a different experimental system to show that expression of these repeats in an endogenous non-transfection system lead to elevated phospho-PKR. And these are mouse embryonic fibroblasts generated from 2 of our BAC transgenic lines that lead to elevated levels of phospho-PKR. And in a liver assay, in an in vitro assay, we show that C9 500 mice also have elevated phospho-PKR. And then if we look in the mice by immunofluorescence, and this is done by Xu Guo, you can see an elevated level of phospho-PKR in the C9 mice compared to the non-transgenic littermates, and that's quantitated there. And also in patient tissues, autopsy tissues, we see evidence for elevated phospho-PKR compared to control autopsy tissue. And we then played around with, could we manipulate this pathway to inhibit PKR and affect RAN translation, or to enhance RAN translation. And so we played around with some vectors that were available to overexpress wild type PKR, or to use a dominant negative form of PKR that would inhibit its function. And so if you look at the blot down here and the red box, this is the lane in which we've added the wild type PKR. So we're overexpressing wild type PKR and a CAG expansion construct. And so there's an increase in RAN translation from just expressing a CAG construct to when you add wild type PKR. And that's quantitated here. But there's a dramatic decrease when you overexpress the dominant negative form of PKR. And that's quantitated here with just a really dramatic decrease in RAN protein levels. And that decrease in RAN protein levels is found not only for a CAG expansion mutation, but also for CCUG, CAGG, and G4C2. And so again, the top panel is the RAN translation, RAN protein blot panel. And you can see that on the far right in each of these lanes, the RAN proteins are decreased, and it's quantitated below. So we used another strategy to show that indeed, if you knock out PKR, you also see a similar effect. You see really dramatic decreases in RAN protein levels across CAG expansion, CCTG, CAGG, G4C2, so multiple types of repeat diseases. And if you inhibit PKR with the TAR RNA binding protein, you also inhibit RAN translation. So we wanted to try to tease out what's the role of the PKR pathway versus other pathways in RAN translation. And so we use the PKR knockout cells that don't have PKR and then added back the phosphomimetic EIF2 alpha. So this is then activating the integrated stress response, but without PKR. And you can see certainly that there's an increase in RAN translation compared to the PKR knockout cells. But when you compare it to what's there in the presence of PKR, there's a significant difference. And so there's much more RAN translation, and RAN proteins are elevated when you when you have EIF S51D in the presence of PKR versus in the absence of PKR. So there seems to be an additive effect of PKR. And this is showing that a similar experiment where we have PKR knockout cells, and at this time we're activating the PERK response with vapsigargan. And again, we partially rescue the loss of the RAN proteins that is seen in the PKR knockout cells, but we don't recover it to the same degree as when we had PKR present. And this is ISRIP is the integrated stress response inhibitor, is another strategy to try to inhibit the effects of the integrated stress response. And we get a partial decrease in RAN protein levels, but not nearly as much as we do when we target the PKR pathway. So I'll go through this pretty quickly. We have a mouse. We use the dominant negative PKR in an AAV strategy to test if inhibiting the PKR pathway in vivo would improve behavior in the mice. And these were P0 intraventricular injections of female mice, and the mice were assessed at 3 months of age. And at 3 months of age, the GA aggregate number went down in the PKR K296R inhibitor treated mice. And that's quantitated here. And also the GP levels went down. And the open field abnormalities that are seen in the C9 mice, where they prefer the extremity or the outer edge of the cage, are reversed in the dominant negative mice. And there were 9 of 15 digigate parameters that were, again, normalized in the mice. And so you can see the non-transgenic and the C9 mice have differences, and these differences are corrected with the K296R treatment. So then we had a really exciting visit from Nahum Sonnenberg, who was talking about metformin and its use in Fragile X syndrome. And I was listening to a seminar, and I was thinking, boy, if metformin could work against RAN translation, that would be fantastic. And so metformin ameliorates core deficits of in a mouse model of Fragile X syndrome, and Fragile X syndrome is thought to have translation problems. So metformin improved the phenotypes there, sorry, hey, Bert. Anyway, metformin improves survival and behavior phenotypes in Huntington's disease, and RAN translation in Huntington's disease is also a factor. So we were really excited about this observation that Nahum Sonnenberg had, that metformin works for Fragile X syndrome, but it's thought to work in a different way. And we were eager to try metformin because it improved the phenotypes in Huntington's disease. And there's RAN translation in Huntington's disease. So we see this decrease of metformin, of RAN proteins in metformin treated cells. And you can see that when we treat cells, we see this decrease in GP levels, in LPAC levels, in alanine levels, and that's quantitated here. So in cells, we saw a decrease, and so we were really excited and really surprised. And then in cells that have this metformin treatment, we also saw that the levels of phospho-PKR were decreased. And metformin reduced the levels of RAN proteins in the treated mice. So we treated them with metformin in the drinking water at 5 mg per mil, beginning at 2 months of age. And that led to a decrease in the GA protein, a decrease in the GP protein, and again, improved behavior by digigate and improved open field behavior, decreased neuroinflammation, and really importantly, improved motor neuron survival. And so in summary, then, we have a model in which PKR activation is a driver of RAN translation, and the expansion mutations mimic the viruses and activate PKR, and that metformin, TRBP, PKR K296R are all really powerful ways to decrease RAN translation. And RAN translation can be activated by EIF2 alpha phosphorylation, but it doesn't seem to be the only thing. There seems to be an additive effect of PKR that's independent of EIF2 alpha. But once you get RAN translation and RAN proteins, you can have misfolded proteins that go back and activate this integrated stress response through PERK and activate EIF2 alpha, as Green and Todd suggested, which starts a feed-forward loop. So to conclude this part of the talk, repeat expansion RNAs activate PKR and increase RAN protein levels. Blocking PKR reduces RAN protein levels. PKR inhibition with AAV, PKR K296R, or metformin decrease RAN proteins and improve behavior and pathology in the mice. And metformin is a novel phospho-PKR inhibitor. Metformin treatment and PKR are really promising therapeutic strategies for C9orf72 ALS, and perhaps a growing number of repeat expansion diseases. And the really exciting thing for me about metformin is that it's a cheap FDA-approved drug, and we've actually started a clinical trial to test the safety of metformin and potential efficacy to reduce RAN proteins in C9 ALS mice. And this is done in collaboration with James Weimer, Emily Plowman, Tim Garrett, and David Valancourt. And we're looking at a combination of things, including the neurologic exam, speech and swallowing, metabolomics, and imaging, and of course, RAN protein levels. And I want to end by thanking many people who have been involved in this work, and those involved in recent work in the lab and past work in the lab, in recent past work are listed here. Tao Zhu has discovered RAN translation, and it's been a great pleasure to work with him for many years now. And Leanne Nguyen was responsible for the antibody work, and then Tao Zhu, the metformin work and the PKR work. And we've had great collaborations with Nahum Sonnenberg, also with Fabio Monterasio and Jan Grimm from Neuroimmune, and other people at the University of Florida, Johns Hopkins, Biogen, University of Bern, and University of Rochester. And I thank the funders, and thank you for your attention, and I'd be happy to answer any questions.
Thank you very much. Very exciting, great talk. I just want to hear your opinion about the GR RAN translation products. Like there's a lot of data in cells in particular that they're like toxic.
Right.
But you're not observing them, or you, not that you're not observing them, but most of the mention is about the GA. So what is your, like, all these balance between the different?
Well, we think that our evidence in the mice is just that targeting GA improves the phenotypes, but targeting GA reduced the GR proteins. So and the GP proteins. So it probably has this additive of beneficial effects. So we're targeting GA, but GA has been shown by cryo-EM to trap proteasomes. And so it probably forms this network that's really bad for protein homeostasis. And by getting rid of GA, we're reducing GR. We're improving protein homeostasis. We're improving proteasome function. And so we think that we're, and so I think I'm agnostic to which is the worst RAN protein, but I think our results clearly show that you can target GA without disrupting or changing the RNA foci or the RNA levels and improve disease. So by targeting GA, you're targeting RAN proteins, decreasing them, and that improves the phenotypes. So it could be GRs driving disease. I'm agnostic to that point. So.
Yeah, okay.
But I agree, there's a lot of support out in the literature for super toxic features of the arginine proteins, PR and GR.
And just before we go to the other questions, do you think that they co-localize and co-aggregate the GAs kind of across eating, or you think it's more that the GA purely traps the proteasome and that's a possibility for? Do you think I did together or?
Up there.
Hello? I think you froze.
Hello?
Yeah,
Can you hear me?
Yeah. You were frozen for a minute.
Okay, sorry about that. So can you repeat the question? I'm not sure I
was asking whether they co-aggregate and can cross, like for example, GA can cross or co-localize and cross-seed one another. Do you have an evidence of that?
I think there's some evidence, but there's a lot of also evidence that they're not always co-localized. So, but it makes sense that if you're improving proteasome function by freeing up the proteasomes that are trapped by the GA proteins, you're going to have an improvement in protein homeostasis and protein turnover. But it could, certainly the antibody effects can, in addition to being intracellular, could be extracellular as well.
Yeah, thank you. Let's go with the Q questions. I don't want to occupy much of your time.
You have two people already in the panel to ask questions, Dave and Christian. Dave, go ahead.
Oh, thanks. It's very fascinating talk, in fact. But I'm going to may I ask you two questions out of ignorance. The metformin is also prescribed for people with type 2 diabetes. And now it's exciting to know that it has some effect on ALS as well. What is the connection, if there is anything, between the activity of metformin with type 2 diabetes, which involves probably different proteins, and the ALS that you mentioned?
Yeah, I mean, I think metformin has a lot of different effects. It's a very small molecule. It's not. I'm not sure that the pathway for type 2 diabetes has anything to do with its effect on PKR. Probably not, but type 2 diabetic. Well, let's see. I should say it this way: metformin has been used as a first-line defense for type 2 diabetes for many, many years globally. And so it's got decades of efficacy data, and it's very inexpensive. So it's used globally. And there have been some studies that have shown that type 2 diabetes seems to be protective for ALS.
I see.
But there isn't yet, we've shown these effects in mice, so we don't know if this is effective yet in humans. We're doing a trial to see if it's safe to give to C9 patients and if it has any effect on the RAN proteins in vivo in humans. But we're excited by it, but we don't think it has anything to do necessarily with with its effect on diabetes.
May I ask you one other question? In the last diagram, you showed some interaction between PERK activation, which, as I understand it, is something in response to the unfolded protein response. And so that pathway intersection also is something that confuses me, how that happens.
How PERK would be activated?
Yeah.
PERK would be activated. The model is that it's the RAN proteins that misfold, and they're just like aberrant proteins that have these repetitive amino acids that form aggregates, and that leads to ER stress.
But then there are some chaperones that are involved to control that stress, right?
Yeah, the proteins are not necessarily pure proteins. So they have C-terminal regions that go until the stop codon in each frame. So it's not well understood yet exactly how that works.
Thank you very much.
Well, Christian, you can ask question directly.
Okay, thank you very much. I have a follow-up question on the metformin question that was asked already. Now, you mentioned that type 2 diabetes seems to be protective for ALS due to this expansions. I was wondering, in principle, that one should see a difference between metformin treated versus maybe the metformin treatment should then rescue the people. From ALS. So are all the people that you mentioned that type 2 diabetes protects them from ALS, are they all under metformin treatment?
Well, so those are that was a population study, so it wasn't a study that looked at C9 patients at all. It was just a study of type 2 diabetic patients, and that they were protected. They seem to have lower incidence of ALS, and the most common drug treatment for type 2 diabetes is metformin. And so more work needs to be done to see if it's really, there's a relationship between metformin and ALS. So that's a missing piece. And but I think that the data out there that there seems to be an effect in multiple studies where the type 2 diabetics seem to have a protective effect against ALS is interesting. You know, C9 ALS is a common type and the most common type of ALS, but it's only about 10% of the sporadic cases and familial cases. So it's maybe there are additional repeats out there that cause ALS, but there's a lot to unpack there. It's not clear that metformin does have an effect on patients, on patients through the RAN translation mechanism yet, but there is this epidemiological data that's worth following up on.
Thanks a lot.
Hi, I was wondering if there are examples of native RAN translation functions outside of disease states. I wasn't sure if there are regions in the genome that are repetitive that it could be happening with.
Yeah, Peter Todd has some really nice, I don't know if Peter's on here, he could answer that better than I could, but he has some evidence that in Fragile X tremor ataxia syndrome, there is a upstream there's an upstream open reading frame that may impact the downstream open reading frame, and that could have that there could be a normal role for the repeat in that. The human genome more broadly though is highly, filled with repetitive sequences, and it's not yet clear how many of those repetitive sequences are involved in disease and how many of them undergo RAN translation. We found RAN translation by looking at the repeat expansion in disease, and that's primarily, what we've been looking at ever since. But I think there's a lot of work to be done to understand what the normal biology of all these repeats are, and but it's hard to imagine that there's not a normal role for RAN translation.
Thanks.
Long Chang, we can ask you a question.
Yeah, that's fantastic talk. Thank you so much. So my first question is, so your mouse model, I'm very interested in. Also, I'm interested in FTD. So the question is, for your mouse model, do they have like the FTD phenotype, like a cortical neuron degeneration? Also, whether or not your antibody also can reach to the brain or hit the upper neuron, or the area in the neuron in the brain responsible for the IPD. And that's my first question. Yeah.
Yeah, the antibody gets into the brain, and so there's co-localization in the neurons that have the aggregates in the brain.
Oh, that's great.
Yep.
Yeah. And the second thing, all your in vitro work done in the cell line or in the lung from the mouse model, and do you ever test in the C9orf human iPS cell line induced neuron? Thank you. That's my last question.
Yeah, we did some iPS experiments on the antibody in collaboration with Jeff Rothstein's group, and the antibody reduced the RAN proteins in the iPS cells, in the C9 iPS cells there as well.
Awesome. Thank you.
Yeah.
Bina, sorry. Yeah.
Oh, shall I ask?
Yeah. It's your turn, Bina, please.
Okay, thanks. Thanks for that exciting, discovery. I wasn't personally aware of it. So I have a little more fundamental question from a protein translation point of view. So when you have other non-ATG codons, but in those cases, you still initiate the synthesis with the methionine, right? But in this particular case, the right codon is charged and then the translation continues, right?
Yeah, so we have a mass spec evidence that the protein translation in the alanine frame can begin with an alanine, and with an alanine you can have a methionine that's removed, but we don't think that the codons are permissive enough to allow an methionine initiator tRNA to possibly fit into the site to initiate translation. So you can, if you have alternative codons, that are close to an AUG initiation codon, then canonical like mechanism, non-AUG like mechanism can be used in the same pathway, same ternary complex, but with when the sequence doesn't have anything that looks like an initiation codon at all and is really different, then it is initiating in a different way.
So I have a couple of questions, right? One, is there a different initiation factor when you initiate such translation? And the second question is, do we know, or does your group know what is the significance of the upstream 5' region sequences to initiate translation in such a scenario?
Well, yeah, there are different initiation factors that have been reported. So there's 2A and 2D that may play a role in RAN translation. But there's no, every every situation is different. So there are about 11 different diseases now that have RAN proteins. The upstream sequence is really important. And there are examples in C9 where an upstream CUG initiation codon, so that's an AUG-like codon, is really important for initiation of one of the reading frames, of the protein translation in one of the reading frames. And but in some cases, and we've experimentally done this, we've changed the upstream regions, and they just behave differently. So some involve a cap and some do not involve a cap. And some initiation factors are important in some frames and not other frames. So it's going to be disease and frame specific. But I think in general, you can have an AUG there. Like in Huntington's disease, there is a large open reading frame, but that doesn't prevent the alanine or serine from being made across the same CAG expansion transcript. In other cases, you have a expansion mutation located in an intron that's probably spliced out, and it's just not clear. I mean, that there can be a variety of different sequences that can still support translation. So it tends to be more efficient if there's an AUG-like initiation codon under normal conditions. But when you stress the cells, then this non-AUG really repeat dependent translation can get more of a foothold.
So wouldn't you be concerned about the regulation part of it? Because if it is going to be, so non-specific, then wouldn't the cell have this disadvantage of, having a much more tightly regulated protein translation?
Yeah, we think that targeting PKR is a good strategy because there's a PKR knockout mouse that does pretty well, but certainly there could be off-target effects, and that needs to be looked at carefully. But we think that there's opportunity to target that pathway because, like many drugs, there's some liability, but if there's this chronic expression of a repeat protein and you just need to turn it down to get a beneficial effect, we think that there could be a really nice sweet spot where you're not completely inhibiting it, but you're just turning it down. So if it didn't accumulate at such high levels, maybe you'd give people more of a chance to have their protein clearance mechanisms work and clear out the RAN proteins that are accumulating.
Thank you. Thank you.
Thank you very much. Rams, John, do you have any questions? And Bikash?
Well, Laura, that was a great talk. I'm a beginner in this field. I'm not familiar with most of the biological side, but we're working on the dipeptide repeats, myself and Magda's group. I wanted to ask you the question, continuing on the question what Magda asked about the co-aggregates. You mentioned that GA is the most important protein for targeting, and your antibody seems to be working very nicely. What about for other dipeptide repeats and for the co-aggregates?
I didn't say that it's the most important target.
Yeah,
I said that it is, you can target it and it has collateral benefits. And we think that's because of the nature of the GA aggregates, that they trap the proteasome. And if you can increase proteasome function, you can improve things. But targeting GA reduces GR and GP. Targeting GP did not improve any phenotype. So we don't think GP is a good thing to target, but we're agnostic about saying it's the best thing to target. We've only tried to target GA and GP. GA is way better than GP. GP did nothing, but we've never tried to target GR or PR. And so we don't have any information about that.
Got it. Okay, thank you.
And what about the antisense dipeptides. They're much lower levels.
Yeah, they're at lower levels, and we were not able to measure them in our mice, so we don't know what the effect was on the PA or PR levels, but it may have been similar, but we just don't know. They're not expressed at high enough levels to be able to measure them. GP is expressed from both the sense and the antisense direction, and GP goes down, but that's an indirect measure of what is truly antisense and what's sense.
Yeah, and the other question which I have, it's really impressive that you find this antibody which can cross cellular membranes and can target the aggregates. Have you thought of doing the structure of that antibody with the dipeptides? This is something.
Yeah, I mean, Biogen has Neuroimmune licensed it to Biogen, so they're doing all kinds of stuff.
Okay.
But we're not working on it anymore. So but I'm hoping that it will go into, that it'll be useful for, that it'll go into clinical trials eventually.
Yeah. Cool. Yeah, it's quite
but there's
exciting.
Absolutely a lot that could be done.
Yeah, that structurally can be done quite a bit, especially with monoclonal antibody, and that's a ton of stuff which can be done in that. But yeah, hopefully they're working on that, because you can do a lot of rational drug design.
I'm sure they're working on it.
Yeah. Yeah. So one thing which I must have missed, you mentioned it, that it was created in humans, the antibody. It's a little bit more it was not like a little bit more history about like, if you can remind how the antibody was made.
Well, in general,
for that particular case, you're like the one which reported your antibody.
Neuroimmune made it, so I'm not sure about all of the details, and they're a company, so they don't provide all of the details. In general, that they collect blood samples from super healthy agers, and then they produce the antibodies by stimulating B cells with antigen, and then they make recombinant, then they get monoclonal antibodies that they generate, and then they clone them, and then they can manipulate them. But they like this approach because the theory is that they're human antibodies that are beneficial because they're found in super healthy agers. So this group of patients, or non-patients, super healthy agers that they have are like 100 years old and healthy. They're walking around, they have super genes, and the theory is that they have super antibodies. And so why do they make any antibodies to GA at all? I don't know. So do they have long GAs? Maybe there's short GA epitopes, or maybe they do have long GA proteins, some of them. And but these are all samples that are pooled. They don't know on an individual level who this came from or anything about these people as far as which people produced which antibodies. But in that cohort of patients, they were able to isolate a human antibody, multiple GA targeting antibodies and GP targeting antibodies. And the theory is that these super antibodies are beneficial in some way to these super healthy people.
Yeah, that's really that's a really good idea. I mean, just bringing it to the next level, because there is something in these people which we need to turn around and look into it. So it's really great. So you've tested their other antibodies, I assume, GP.
We tested 3 antibodies, 2 GA antibodies and 1 GP antibody.
No GR.
No.
Okay. Yeah.
We didn't find a GR or a PR to look at that met our criteria of being a good antibody.
Yeah.
That would detect things in vivo and, but they may have them now. I don't know. That was some years ago.
Yeah, it's really, really exciting. And it's, again, it's still so fascinating that we have all this translation going on and we even don't know. Do you think that's again, like my last series of questions, do you think it has to do something leftovers from like low-level organisms, all these expanded DNA sequences, which are the culprit of these like repeats? Or you think these are like introduced by some parasites or viruses? Do you have any theories about the like origins of them? I know that you got a similar question, but like, do you think that they are viral origin or?
Yeah, I mean, I think a lot of repetitive DNA is retroviral origin to begin with. Most of the even mice have much shorter repeats than humans do for every given locus that can turn into a disease expansion locus like SCA1. The mouse version of the gene has a very short repeat, and that's true for Huntington's as well and other repeat disorders, expansion disorders. And then in humans, the normal repeat range is quite a bit longer, and that's so that even the normal range is about 30 in SCA1 versus 2 in the mouse. And so that normal length of repeat is thought to have a role in regulating, and fine-tuning the regulation of its function. But that gives the humans some advantages, but some liabilities in the disease area, because the DNA repair enzymes can misread those repeat expansions and get lost and confused and make them longer. And then that leads to an repeat instability and then a predisposition to disease, which can eventually take off and cause these diseases that run in families.
Yeah, that's and we have a long wants to ask a question, but I just kind of want to follow up with a question. This is something I find struggling quite a bit in the literature, since you already mentioned it, because the reports over the length of the repeats are all over. It says that like if you have it in the thousands, and this is in the case of the C9. If you have it in the thousands, for sure you will develop the disease. But there are also reports of a shorter disease in like maybe I've seen 70 or 80, and they've been also reports, pathology reports finding like the run products, let's say in some cases of Alzheimer's disease. Do you think that this shorter disease is kind of coincidental more rather than?
Well, I think it's probably when the shorter repeats are measured in blood, right?
Mm-hmm.
And
these were more like not
brain. And there's a lot of somatic instability in these diseases. And so depending upon the level of somatic instability, these patients with 70 repeats in the blood may have thousands of repeats in some cells in the brain. And the repeat instability, somatic repeat instability, is, I think, hugely important in disease development. And that's really come to bear in the Huntington's field as a big modifier of disease, repeat instability. So if you can stabilize the repeat and prevent it from expanding further, 70 might be fine if you could keep 70 throughout your body. But if you've got thousands popping up of repeats popping up into some cells, and we know repeat expansions are going to make RNA gain of function effects worse, and they're going to make RAN protein expression go up, that could be enough to kill that cell. And so you may have this situation where repeat instability is a big driver of disease because it causes individual cells to become quite vulnerable because their repeats are way too long and they're making too much of RAN proteins. And so it's not really 70 repeats at all that's causing disease. It's something completely different going on in the brain.
For the run translation, when you have like poly-GA, do you have any estimate? I assume it's a range of sizes. Like let's say you have 1,000 of repeats of octonucleotide repeats, right? But like in the run translation, there is a certain breaking point to King. Do you have any idea of the sizes of the run translated peptides, or this is something also?
No, we don't. In vivo, we don't know, but we do know that the repeats can be quite long, and they seem to, the RAN translation seems to get through the repeats in cells for repeats of 100. So we've done that experiment where we put 100 repeats and we have an N-terminal tag and a C-terminal tag, and it gets through the repeats just fine. It doesn't start in the middle or stop in the middle. It's, it goes right through. But well, it might start in the middle, but it goes through to the C-terminal, and that repeat doesn't prevent translation from happening. We also know the C-terminal antibodies work to detect the proteins in the human patients, and they work quite well. So we don't think that there's an impediment to getting through these big repeats to make RAN proteins.
Yeah,
but it's really hard to know. You can't do mass spectrometry on these huge repeat proteins, and no, but lots of people are studying that.
Yeah, thank you. I think Long, you can ask your questions now.
Okay, thank you. So my question is, think about the sporadic ALS FTD. So what's your thought about either you can target on this AUG in this genetics, or general ALS FTD that have common mark like a TDP43 aggregation. Can you, what's your thought if you target on TDP43 aggregation or you target AUG, they're gonna different, or which is what advantage is disadvantage if you think about more sporadic ALS FTD pathology? And so that's my first question.
So let me get your first question before you pile up questions.
Yeah.
So your first question is, if you have sporadic ALS, what do I think of targeting TDP43?
Yeah, or even for this genetics, if I target on some like this other, I think also genetic has a TDP43 aggregation, right?
Also, what has a
TDP43 aggregation? Is TDP43 aggregation also present in your mouse model?
It's present at the very end.
Very end.
Yeah, very end. So it doesn't seem to be it certainly doesn't occur before the RAN protein accumulation.
Right, that makes sense if you target on AUG for this this genetic forms ALS FTD. But for sporadic, do you think if I target on TDP43 aggregation,
what do you mean target AUG?
Oh, the run.
Oh, okay.
Run, because this run also because a lot of people think the TDP43 play an important role in the pathology. So I'm thinking in addition to targeting on this run, do you think also it's reasonable also targeting on TDP43 or combinant together?
Yeah, I mean, I think the jury's out on that. It's certainly worth a try. We have to have multiple shots on goal. TDP43 pathology seems to be a common downstream thing, and also an upstream proximal problem for those that have genetic forms of TDP43 ALS. But I don't know. I am, I think for C9, I think it may be better to target something more upstream. And I think the RAN proteins are more upstream than TDP43 pathology. And that if you have lots of RAN proteins and you correct TDP43 pathology, would that be enough to correct the disease? I don't know, but worth a try, certainly. But I like I think the ASO and more things that are more proximal are perhaps more promising.
Yeah. Yeah. The second one, you already mentioned this run by the antibody. So your TRIM21 indicate they can degrade by the prosome, or by the other data show maybe degraded by the autophagosome or lysosome. In addition to the antibody, do you think also it's worthy to try in the future using the molecular glue, for example, Proteinopathy or Lysotech? People recently developed using the the linker to bridge your run to the E3 ligase or lysosome mechanism to push them to degradation by this the lysosome or prosome using this Proteinopathy technique rather than use the antibody. Because one antibody thing is you have to concern about how cross the BBB or the concentration. And do you think that will be another option using this, the molecular glue to link the run to lysosome, autophagosome, or Proteinopathy?
I mean, I think that's a great idea. If you can get rid of the RAN proteins any way you can get rid of them, I think that would be a good idea.
Okay, thank you. Thank you.
Thank you so much. We really enjoyed it. I don't know, Rams, do you have any more questions?
No, and it's great talk. A lot of good discussions.
Is there anything? So have we gone through the chat things, or?
Yeah, we've gone through all the questions.
Okay.